And now for the star, perhaps, of today's show, which is our newest investment. It's a company called MigVax. We're joined by David Zigdon, who is the CEO, and Professor Itamar Shalit, who is a member of the Scientific Advisory Board and a specialist in infectious diseases. MigVax is leading the charge, certainly here in Israel, and we think globally to a certain extent, in helping us find a path to a successful vaccine. It turns out that this company has been working on a similar project for years before the COVID-19 crisis. As soon as they heard of the crisis breaking, they began to adapt their research to this crisis, and we have made a decision and announced it literally today to lead a $12 million investment round in MigVax. We're delighted to have David Zigdon, the CEO. Please, David.
Thank you, John. I'm the CEO of MIGAL. MIGAL is the multidisciplinary research institute located in Upper Galilee. As John said, in the past 4 years, we have been working on vaccine for coronavirus in avian. We have designed a system that can adapt itself very fast when mutation of the virus show up. Now, when we are facing COVID-19 in human, we have the opportunity to use the same methods that we develop for human, the same method to develop a human vaccine. Now I have Professor Itamar Shalit with me. He explain what we are doing. Itamar?
Good evening and nice to be here. We heard a lot this evening about the coronavirus pandemic and its horrendous impact on health, on economy, on our social life. What's very important is to understand that to suppress it and to go back to our normal life, we have to invest in a vaccine that will be effective. Once we have an effective vaccine that could be dispersed all over the world, then would be the time that we can go out of this pandemic situation and go back to normal life. Now, the virus itself, you can see it in the picture. You see the red spikes. These are the S protein. These are the spikes of the virus, and they recognize receptors on the cells in different areas in the body and cause the disease. Inside the virus, there are other proteins. Some of them are named N proteins. They are important also in eliciting immune response. Next, please. We have identified over the last 4 years, and actually even more than that, that there is a problem with a chicken coronavirus disease called infectious bronchitis virus. It's a coronavirus very similar to the human COVID, but it mutates very rapidly, very often, more than does influenza virus. And the vaccines that we had at that point were ineffective against the newest mutations. So our scientists decided to have a platform approach to create a vaccine that not only will take care of the newer mutation, but whenever a mutation arises, it can be easily adapted and rapidly adapted to the newer mutation. And we have now a human vaccine sketch of the proteins, and you can see the red, the yellow, the gray. These are the S and 2 N's antigens of the virus, and you see that they are linked to a carrier protein, and this carrier protein is extremely important simply because it enhances the immune response against each of these proteins. And what happens is that based on the IBV vaccine, we are now creating a human homologue of that is very, very likely to work similarly to the IBV vaccine, which the results of it I'll show in a second. But we are moving ahead in activating the 3 arms of the immune system. The mucosal arm, which takes care of our respiratory system, our guts, etc., the antibodies in our blood, and also the cellular immunity, which means lymphocytes that can kill cells that harbor the virus. Next slide. Now, this is a key experiment that was done in the Israeli Veterinary Institute. We have looked at the efficacy of the chicken vaccines in chicken, that we gave them the vaccine and then we challenged them with a massive inoculum of IBV virus, which was live and virulent, and looked at the level of harboring of virus after they got the vaccine. And those who did not get the vaccine showed 100% shedding of virus from the respiratory tract and from the gut. What was most alarming is that when we used the commercial avian vaccine that we used at that time, we did not see any reduction in the amount of virus that was shed from the respiratory tract and the gut. But if you look at the right-hand side of the graph, you can see that with our MigVax, after 3 days, we already saw about 15–20% reduction in harboring of the virus. But within 6 days, 80% of the chicks did not harbor any virus. And those who did had a very, very, very low number of viruses. Remember, they got massive inoculum, much higher than what occurs in real life situation. Next, please. Now, people think, hey, the winner takes it all. The one who gets first to the market is going to win it all. No, not at all. The GSK CEO said that we'll need more than one vaccine, and I can attest to you that we're going to be much more than one. The needed amount and the fact that we are going to need it not only to the Western world, but to Southeast Asia, to third world, etc., we will need definitely different kind of vaccines. Next, please. Now, if we look at the competitive landscape, and you can also skip to the next one already, the idea behind every single technology to create vaccine is some way or another to present mainly the S protein, the outside protein, to our immune system. You can do it with a killed virus. You could do it with proteins alone. You could do it with adenovirus that present it. You can do it with RNA that encodes the S protein in our body. Whatever the case is, our immune system sees this antigen and produces antibodies against it. The thing is that it is not clear at all that having antibodies only against the S protein is enough. It might not be enough, and there is also some problems with having only antibodies that may enhance the disease, which is beyond the scope of this discussion. The timeline of the project is the synthesis of the human oral subunit vaccine, the 3 proteins linked to the carriers. These are ongoing as we speak. Again, with all the human structure of the human COVID virus. We are going to start doing safety and efficacy in rodents, in this case, mice, and later on in rats, in May 2020. And the phase 1/2 clinical trial will start hopefully on summer 2020, because we are going to have all the data from the animal studies, and that will be sufficient to create this kind of protocols to the first in human. And with the first in human, within 6 months, we definitely will have data that will help us understand both the safety and the efficacy of the vaccine. And then we will try now to go to the regulatory authorities, and following this kind of first in human studies, design the phase 2B studies, etc., etc. Now, the market, what's the market? The market is 1 billion of those that will be required worldwide in order to adequately address the coronavirus epidemics. We'll need multiple sources in vaccine, and manufacturing will be necessary in many, many, many places. We will not be able to support the need of the whole world. And we know that the vaccine price range from $9.5 to $200. This is the estimation of the American CDC. And cost of goods is estimated as low single-digit USD, and that's our vaccine. And move to the next slide, please. So to summarize, we have an orally based vaccine. We are giving it by mouth. We are not injecting it. It's safe. It's simple. It's less costly. so we can give it without having a nurse in place to give any injection, for example, drops in the mouth. It is based in preventing infection. Most companies moving ahead did not have this data before designing the vaccine or starting phase 1. We already have this proof of concept of efficacy. And we activate the 3 distinct immunological pathway, which is possibly the best way to try and have an efficacious vaccine against this virus. Safety is clear because it cannot cause a disease. It's not a virus. It's not a live virus. It's not an attenuated virus. Sorry, go back, please. There are less chances of side effect. And from a market size, we have an advantage in large-scale manufacturing with low cost. It's produced in an E. coli bacteria, very simple, in a fermenter, very simple. We can create huge amount of doses with relatively intermediate and large-size fermenters. And I think that that cost effectiveness is something that will make us very competitive in the market. And of course, we can adapt to variant viruses because that was the original reason why we started to have this vaccine, because we had to address variation in the coronaviruses. Thank you very much.
Thank you very much, Itamar.